Welcome everyone to today's lesson on respiration, and welcome back to everyone following our weekly lessons, and especially those of you who joined me last week in lesson 1 of respiration. For those of you who didn't join me last week, I do recommend you go back and study that lesson before listening to today's lesson. So we took a deep dive into the fundamental processes of glycolysis, link reaction, the Krebs cycle, which are the opening 3 stages of aerobic respiration. And those 3 processes not only are capable of generating their own ATP, but then they set the stage for the final act in aerobic respiration, which is oxidative phosphorylation, which we are going to cover today. Not only are we covering oxidative phosphorylation, we're going to look at how you measure the rate of respiration and also look at anaerobic respiration. But first, we're going to start with looking at the primary output that we got in previous 3 stages of respiration that we studied last week, and that is NAD and FAD, or reduced NAD or FAD, and this is basically going to be the input into oxidative phosphorylation. So we're firstly going to look at today the critical role of these coenzymes in aerobic respiration, but what exactly are they and why are they so important? So NAD and FAD are basically coenzymes that function as hydrogen carriers in the process of respiration. These coenzymes play an instrumental role in the transfer of hydrogen atoms during respiration. So remember, in the Krebs cycle, at times we kept losing hydrogens and they were taken up by the NAD and the FAD, or they were reduced. So a hydrogen atom consists of a hydrogen ion, a H+, and an electron. And when NAD and FAD gain a hydrogen atom, they become reduced. And an easy way to remember this is the acronym OIL-RIG. And what that means is oxidation is loss and reduction is gain. So in relation to FAD and NAD, when they gain an oxygen, they are reduced. So it can be a little bit confusing because they gain in something and then reduced kind of suggests that you are losing something, which is why it's helpful to remember the acronym OIL-RIG. Oxidation is loss, reduction is gain. So as I said, these coenzymes are like the couriers for hydrogen atoms. So they're constantly gaining a hydrogen, losing a hydrogen, and they're just basically used to transport the hydrogen from one place to the other during respiration. So they accept hydrogen atoms from various stages of respiration and then transport them to a crucial location somewhere else. The electron transport chain on the inner mitochondrial membrane is where they transport the hydrogens they pick up from the Krebs cycle to. And it's worth noting that when hydrogen atoms are removed from NAD and FAD in the electron transport chain, we then refer to it as being oxidized. So oxidation is lost. So when they lose a hydrogen, which we will learn about coming up in the lesson, they are oxidized. And let's remember, the whole point of respiration is to actually make ATP. And these hydrogen ions and electrons have a pivotal role in the electron transport chain, which is going to be the biggest generator of ATP during oxidative phosphorylation. It's the final phase of respiration. So the electrons derived from reduced NAD or NADH and FADH2 are delivered to the electron transport chain. When the electrons are lost through the electron transport chain, hydrogen will be released. And we will go through this in much more detail shortly. But just to sum up, coenzymes NAD and FAD are carriers of hydrogen atoms. So they carry the hydrogen atoms from the Krebs cycle to the electron transport chain. And their reduction and oxidation, this OIL-RIG acronym, plays a pivotal role in the synthesis of ATP. So remember, oxidation is loss, losing hydrogen, and a reduction is a gain in a hydrogen. So let's recap now that we fully understand how important these NADs and FADs are, the sources of the reduced NAD and FAD that we got from our first lesson. So from glycolysis, we obtained 2 reduced NAD molecules, or 2 NADH molecules. In the link reaction, we also got 2 reduced NAD molecules. From the Krebs cycle, we actually produced 6 reduced NAD molecules and additionally 2 reduced FAD molecules. So this is a lot of hydrogen being transported from these first stages of respiration that we covered last week into oxidative phosphorylation, the electron transport chain, and ultimately into ATP synthesis. So moving on to the next phase of our lesson, which is going to be on oxidative phosphorylation and ATP synthesis. And here we're going to slowly unravel what oxidative phosphorylation is as the final stage of aerobic respiration and how it contributes to the production of ATP. So oxidative phosphorylation is a crucial step in aerobic respiration that takes place in the inner mitochondrial membrane. It plays a central role in producing a significant number of ATP molecules and results in the creation of water from oxygen. And the current model for understanding oxidative phosphorylation is the chemiosmotic theory. And the chemiosmotic theory essentially postulates that the energy generated by electrons passing through a chain of proteins in the membrane, known as the electron transport chain, is then used to pump hydrogen ions up their concentration gradient into the intermembrane space. So let me just repeat that again. So picture the inner mitochondrial membrane. You have proteins embedded within that membrane. And the chemiosmotic theory postulates that as an electron is passed through a chain of proteins embedded in that membrane, that passing of energy is then used to pump protons or hydrogen ions up their concentration gradient into the intermembrane space. So hopefully you are watching the slides and you can see this cartoon of the process. And what the cartoon is showing is firstly the electrons being donated from the NADH and the FADH. And you can see when the electron is donated to the first protein in the electron transport chain, that the electron has been donated to the protein, and then you're also getting a release of hydrogen. So that is where those hydrogen ions are then accumulating in the matrix, ready to be moved from the matrix into the intermembrane space. Now, you know from your previous lessons that for something to flow against their concentration gradient, you need a protein pump. So these electron transport proteins are actually also hydrogen pumps that will move the hydrogen ions from the matrix with the lower concentration into the intermembrane space that actually have a higher concentration of hydrogen ions. This then contributes to an increase in concentration gradient. And then we also finally have a hydrogen proton channel. And in this channel, hydrogen is going to flow from high concentration to a low concentration. And this proton channel also doubles up as an ATP synthase, meaning it will make ATP. So as we're getting a build-up of hydrogen ions in the intermembrane space, they'll then start to flow more and more through the proton channel down its concentration gradient through the ATP synthase channel, and ATP synthesis will occur by phosphorylating ADP into an ATP. So the energy harnessed from the movement of the hydrogen ions down their concentration gradient by facilitated diffusion drives the phosphorylation of ADP to ATP by ATP synthase. And this is where all the ATP synthesis happens. So before I go over again, just a quick summary. Oxidative phosphorylation is a critical process in aerobic respiration that involves this chemiosmotic theory, where the energy from the electron transport chain is used to pump protons up their concentration gradient, then in turn they move down their concentration gradient, generating ATP from ADP. So let's go over it one more time, slow down a little bit. So we have reduced NAD and FAD molecules from the first 3 stages of respiration. These reduced NADs and FADs serve as donators of electrons to the electron transport chain. So the electron transport chain is composed of a series of membrane proteins and electron carriers. They are strategically positioned close together, allowing electrons to jump from one carrier to the next. So the electrons are donated to protein number 1 in the electron transport chain, and they move from one to the other. Now, the other important thing you need to remember about this inner mitochondrial membrane where these electron transport chain proteins are positioned is that it is impermeable to hydrogen ions or protons. So these electron carriers are then going to play a role in pumping protons across the membrane. Remember, each time an NAD or an FAD donates an electron to the electron transport chain, it is also losing a hydrogen ion. So the inner mitochondrial membrane's impermeability to protons necessitates the presence of these electron carriers to actively pump protons across. Remember, it's also going up its concentration gradient, so it needs to be an active pump. So this then creates a concentration gradient where protons are moved to a more concentrated intermembrane space compared to in the mitochondrial matrix. What then happens is there's a flow of protons back into the matrix down its concentration gradient through diffusion, facilitated by a channel protein or ATP synthase. It is the movement of protons down their concentration gradient through this ATP synthase channel protein that provides the energy required for ATP synthesis. And the ATP synthesis happens by phosphorylation of an ADP into an ATP. And then the final thing you need to know for oxidative phosphorylation is that oxygen is the final electron acceptor. So as the electron moves down the protein carriers in the electron transport chain, the final recipient of that electron is going to be an oxygen molecule. And then oxygen combines with protons or hydrogens and electrons to form water, effectively concluding the electron transport chain's vital role in the generation of ATP. And just to relate it back to the bigger picture respiration equation, obviously we know that H2O is a side product of respiration. And this is also important in terms of comparing aerobic to anaerobic respiration. So aerobic is dependent on oxygen. So for aerobic respiration to happen, you need to have that oxygen present at the end of the electron transport chain to receive the electron to conclude aerobic respiration. And that moves us on nicely to our next section, which is going to be on anaerobic respiration. So when oxygen is sparse, cells must adapt. So what happens when there's little or no oxygen available for respiration, and why is anaerobic respiration therefore important? So when cells find themselves in an environment with insufficient oxygen, for example, if you're exercising, there are several consequences. So the number one I just addressed is that there is no final acceptor for electrons in the electron transport chain. As a result, the electron transport chain ceases to function. So it can't hold, picture it as not being able to hold more than one electron at one time. If you have a chain of people and you're passing a bowl from one person to the other, you can only hold one bowl at a time. So ultimately what happens if there's no bucket to put the ball into at the end of that chain, the balls are going to start to get backed up until you get to the point where you can't take on any more electrons. So this leads to a halt in the ATP production through oxidative phosphorylation, since you're then getting less hydrogens being pumped into that intermembrane space and coming back into the matrix through the hydrogen channel. The next consequence is then the reduced NAD and FAD are left unoxidized by an electron carrier, meaning they remain reduced. And consequently, there is no oxidized NAD or FAD available for dehydrogenation in the Krebs cycle, causing it to stop. So remember, all of this is happening concurrently. So when the reduced NAD or FAD is donating its electron and losing its hydrogen for the electron transport chain, they then go back into the Krebs cycle to be reduced once again, pick up another hydrogen and come back to the electron transport chain. So when there's no oxygen at the end of the process, everything gets backed up. And the solution to this is anaerobic respiration. So it's a way of the cells to produce at least some ATP in a low oxygen environment. And this process involves pathways that allow cells to continue glycolysis, producing a small amount of ATP. Remember, going back to glycolysis is not oxygen dependent and you can produce small amounts of ATP. And different cells employ various pathways to achieve this depending on their type and the microenvironment they inhabit. So firstly, starting with ethanol fermentation. So yeast and other certain microorganisms will resort to ethanol fermentation, while other microorganisms such as mammalian cells opt for lactate fermentation. So let's start firstly with ethanol fermentation. So in ethanol fermentation, reduced NAD transfers its hydrogens to ethanal, ultimately forming ethanol. So by ethanol fermentation, we just mean that we're going to make ethanol. So the hydrogen on the reduced NAD is transferred to ethanal, forming ethanol. And that occurs in a three-step process. So the first step involves decarboxylation of pyruvate, resulting in the formation of ethanal and release of carbon dioxide. Subsequently, ethanal is reduced to ethanol with the assistance of an enzyme called alcohol dehydrogenase. Dehydrogenase meaning to lose a hydrogen. And then finally, it's important to note that ethanol, once it's formed, cannot be further metabolized. It is essentially a waste product. So pyruvate is decarboxylated, forming an ethanal and a carbon dioxide. The ethanal is then reduced to ethanol by an enzyme called alcohol dehydrogenase. And that ethanol is then a waste product and cannot be used. So next, let's turn our attention to lactate fermentation. So in lactate fermentation, reduced NAD transfers its hydrogens to pyruvate, resulting in the formation of lactate. The enzyme lactate dehydrogenase then plays a crucial role in this process. Unlike ethanol though, lactate can be further metabolized. So after lactate is produced, it can take two different paths. So firstly, lactate can be oxidized back to pyruvate, which can then be channeled into the Krebs cycle for some ATP production. But of course, this process of oxidizing lactate back to pyruvate does involve an extra demand for oxygen, and this is often referred to as the oxygen debt. So this will explain why people and other animals will breathe more deeply and rapidly after engaging in strenuous exercise. Is because you have gone through this process of metabolizing lactate and you now have an oxygen debt which you need to breathe in more heavily, more frequently to regain that oxygen to pay off that debt essentially. And lactate can also be converted into glycogen, which, as you know, is a storage form for glucose, and that transformation into glycogen serves as a vital energy reserve in the liver. So just to recap, we covered two different anaerobic pathways. Firstly, in yeast and certain microorganisms, we covered ethanol fermentation. So ethanol fermentation involves the decarboxylation of pyruvate, resulting in the formation of ethanal and the release of carbon dioxide. And then subsequently, ethanal is reduced to ethanol with the assistance of an enzyme called alcohol dehydrogenase. And in humans and other animals, we get lactate fermentation. So in lactate fermentation, reduced NAD transfers its hydrogen to pyruvate, which results in the formation of lactate. And the enzyme lactate dehydrogenase is what catalyzes that reaction. And unlike ethanol, the lactate can be further metabolized. It can either be oxidized back to pyruvate, so we can keep repeating that process. But remember, as we do that, we will build up an oxygen debt. Or alternatively, lactate can then be converted into glycogen and it's used as a glucose storage in the liver. So for our final section on this lesson, we are going to look at investigating the rate of respiration. So hopefully many of you will have the opportunity to do this in person. But for those of you who haven't, I'll try and do my best at describing how this process works. This is, for many of you, a mandatory practical, but you can still get asked not just in your practical exams, but in your theory exams as well. So essentially in these experiments, we are going to be measuring the effect of temperature on the respiration rate in yeast. So I'll just very briefly introduce a redox indicator. So this is going to be how we measure the rate of respiration. And a redox indicator is basically a dye that we use to indicate colour changes in response to either being reduced or oxidized. And I'll go into that in more detail in a second. But just so you know, that is the readout of the experiment, a redox indicator, which will change colour in response to either being reduced or oxidized. So let's go through the experimental setup. So we'll be working with yeast, which is a microorganism known for its ability to respire both aerobically and anaerobically. However, in our experiments, we will focus just on the anaerobic respiration. Let's recap a little bit about a little bit on the theory from last lesson. So throughout various stages in respiration, dehydrogenation is a common occurrence, so losing a hydrogen. And hydrogens are removed from substrate molecules, and these extracted hydrogens are then transferred to the final stages of aerobic respiration, as we just discussed, known as oxidative phosphorylation. And these molecules that I refer to are NAD and FAD, coenzymes NAD and FAD, and they essentially become dehydrogenized, so take up an oxygen in one place and then move that oxygen to the next stage of respiration. So our redox indicators are introduced into the experimental setup, so into the yeast, and they basically assume the role of molecular sponges for hydrogen. So they're going to take up all the hydrogens they can. So therefore, instead of NAD, these indicators, these redox indicators, take up the hydrogen and then they undergo reduction themselves. And that reduction leads to a significant colour change, transferring them from blue to colourless. So these redox indicators, the two you need to know the names of are DCPIP and methylene blue. So the rate at which this colour changes then will provide an insight into the rate of respiration. So essentially what we're going to do is add this blue dye to the yeast, start a timer, and then you can measure how quickly that blue dye disappears and the solution becomes clear. And that will be indicative that the dehydrogenation is complete. So all of the hydrogen has been removed from the substrate and taken up by the redox indicators, and that is a proxy measure of the rate of respiration. So the rate at which the colour change occurs provides insight into the rate of respiration. The faster the rate of respiration, the more rapidly hydrogens are released, consequently leading to the swift reduction of redox indicators. This relationship means that the rate of the colour change corresponds to the rate at which dehydrogenase enzymes are functioning, and by extension the rate of respiration in yeast. And so to quantify the rate of respiration, we employ the formula rate of respiration per second is inverse to time. And so 1 over time in seconds. And this formula highlights that the inverse relationship between the rate of respiration and the time taken for the solution to change from blue to colourless. So in terms of the apparatus and experimental design, we will need yeast, glucose solution, test tube, stopwatch, and the redox indicator, for example, DCPIP. And I'll just go through the method. So specifically, if we measure the effect of temperature, so we begin by adding a predefined volume of yeast suspension in the test tubes. This has to be the same volume of yeast per test tube. And these test tubes can also contain varying concentration of glucose if you want to measure the effect of glucose, for example. The test tubes are then placed within a temperature-controlled water bath, ensuring the water temperature remains stable throughout the experiment. And then a 5-minute incubation period is crucial to achieve the desired temperature. So to get the temperature from room temperature to the temperature you've set the water bath at. Next, you add a specified volume of DCPIP. So it's not until this stage that you start to measure the actual respiration that's happening. So you're going to put a specific amount of the dye into the yeast. And then at that moment, you start the stopwatch to initiate the time recording. The stopwatch is stopped once the solution becomes completely colourless or loses all its blue colour, and that transition time from blue to colourless represents the end point of the reaction. It's essential to acknowledge that discerning the precise end point of the reaction can be somewhat subjective. So someone in the group may think that it's already turned colourless, but you may want to continue for a few more seconds to be absolutely sure. And that can lead to a little bit of variation within the experiment. It is for that reason that the same person should be assigned to do all your repeated experiments, so they will have the same judgment of when the solution is completely colourless. In terms of other variables and how you control for them, so the volume of dye added, a surplus of dye can elongate the time taken for the colour to change. So if you've added extra dye, that can result in your results being longer, or the time points being longer than you should have been. The volume of yeast suspension, so how much yeast you actually have in there. If you have higher quantity of yeast cells, again, it can artificially inflate the rate of respiration. The type of substrate, i.e. glucose. So it's worth noting that the yeast cells respond differently to different types of substrates. So use a consistent substrate. So essentially just make sure you're using the same glucose every time. And also the same concentration of substrate. So when you buy glucose, it can sometimes be at a higher concentration than in other bottles. Temperature is the variable that we were measuring, but if you are, for example, measuring the concentration of glucose instead, you then need to control for temperature by putting all of your tubes in the same temperature water bath. And finally, pH. And for this, you can employ a solution to regulate the pH levels, ensuring that enzymes, especially dehydrogenases, which is the important enzyme in this reaction, and ensuring that pH is not affecting any of the enzymes within the experiment. So what are your expected results? So you should essentially plot a graph of temperature against time, and you will see that as the temperature increases, the rate of respiration also increases, so the time for the solution to go colourless will reduce. This means that the hydrogens are released by the reactions more quickly, hence the DCPIP accepts electrons and hydrogens more quickly until all molecules in the DCPIP are reduced, and it will take less time to turn from blue to colourless. So the rate of respiration increases as you increase the temperature. So there are some limitations to this experiment. So it's not measuring the rate of the dehydrogenase, so the enzyme that's reducing your NAD/FAD directly. So it is instead predicting what the rate would be by measuring the rate of the hydrogen release from the reactions of respiration. And then the second limitation is, as I just mentioned, that the end of the reaction, the colour change, is subjective, and therefore you should always use the same person to control, to determine when the reaction is finished, to control for any variation from one person's opinion to the other. So congratulations, you made it to the end of these 2 lessons on respiration, which can be really difficult and pretty heavy topics. And just to recap what we covered today specifically, so we looked at the role of NAD and FAD and them being oxidized and reduced, and how they connect the different stages of aerobic respiration and the sources of those reduced coenzymes. Moving into oxidative phosphorylation and the electron transport system. Then we covered a little bit of anaerobic respiration, and then practical experiments to investigate the rate of respiration. So let us know in the comments if you have any questions, and as always, like, subscribe and follow us on our Patreon channel, where we have follow-up lessons specifically doing revision and practicing exam questions on this topic. Bye!
Bye.